You think parasites just live inside their hosts? You're wrong. Some of them are surgeons, precision surgeons. They rewire your behavior. They steer your body. And the most famous one, the zombie ant fungus, does all of this without ever touching the brain. For 150 years, every scientist assumed the fungus invaded the ant's mind. In 2017, a Penn State graduate student looked down a microscope and discovered we were all wrong. The brain was untouched. The body was a puppet. And the death grip we are about to describe. We have a fossil of it from 48 million years ago, older than humans, older than primates, older than you can imagine. Stay with me. By the end of this video, you will see the line between you and your passengers the way scientists see it. And it is thinner than you want to believe. It is solar noon in a rainforest. The air is heavy with water. Cicadas are building toward their loudest moment of the day. And somewhere on a leaf, about 25 centimeters off the forest floor, a carpenter ant is doing something it has never done before. It is climbing down, not up. Carpenter ants live in the canopy. They follow pheromone trails. They forage on schedule. This one has left the trail. It is alone. It is moving at the wrong time of day, in the wrong direction. It stops on a leaf. It crawls to the north side of the central vein. It opens its jaws and bites down, and it does not let go. For the next several hours, the ant clings to that vein. Its legs twitch. Its antennae droop. By evening, it is dead, but its body stays locked to the leaf, because something is still working its jaw muscles, something that does not want it to fall. A few days later, a single thin black stalk pushes out from between the ant's eyes. It grows upward, like a candle on a corpse. At its tip, a sac swells. The sac bursts. Microscopic spores rain down onto the forest floor, onto the trails where the dead ant's sisters still forage. The killer is a fungus called Ophiocordyceps unilateralis. It does not bury the body. It uses it as a launch pad. And the question that haunted scientists for over a century is, how did it do this? The first person to ever describe this fungus was Alfred Russel Wallace. You might not know the name. You should. Wallace is the man who co-discovered evolution by natural selection, the man who, in 1858, sent Charles Darwin a letter from the Malay archipelago that finally forced Darwin to publish his ideas. Without Wallace, On the Origin of Species might still be sitting in a drawer. In 1859, Wallace collected a strange specimen in the Indonesian rainforest, an ant with a stalk growing out of its head. He had no idea this thing was a zombie maker. He just thought it was a beautiful, terrible fungus. For the next 150 years, scientists studied this fungus. They watched the death grip behavior. They counted the species. They measured the spore release. And they all assumed the same thing. The fungus must be inside the ant's brain. How else could it possibly control its movements? It seemed obvious. If you want to control a body, you control the brain. There is just one problem. That is not what was actually happening. To find out the truth, you would need a microscope so powerful it could photograph a single muscle fiber slice by slice, and a researcher patient enough to map every fungal cell in 3 dimensions. That researcher arrived in 2017. Her name was Maridel Fredericksen. But here's the part nobody talks about. Maridel Fredericksen was a master's student in David Hughes's lab at Penn State. She used a new technique called serial block phase scanning electron microscopy. It slices a sample 1 molecule thick at a time and photographs each slice. Then a computer rebuilds the inside of the specimen in 3 dimensions. She fed infected ants into the machine, and she waited. When the images came back, the lab was stunned. The fungus was everywhere. In the legs, in the head, in the thorax and abdomen, wrapped around individual muscle fibers in a 3D network. About 25 strands of fungus per single muscle fiber, all linked, all communicating. The ant was not really an ant anymore. As David Hughes put it, these manipulated animals were a fungus in ants' clothing. But the truly stunning finding was about the brain. The brain was clean, untouched, pristine. The fungus had grown around it, surrounded it, but had never invaded it. Every cell in the ant's nervous system was alive and intact. At the moment the ant climbed down, walked to the leaf, and bit the vein. For 150 years, we had been wrong. The fungus is not a brain invader. It is a body invader. Hughes described it like this. The parasite is controlling host behavior peripherally. Almost like a puppeteer pulls the strings to make a marionette move, the fungus controls the ant's muscles to manipulate the host's legs and mandibles. The brain was kept alive on purpose. The fungus needed the ant's eyes and antennae to find the perfect leaf. It only took over the body once navigation was done. Think about what that means. The ant arrives at the leaf fully aware. Its senses still report. Its brain still fires. And then its own muscles refuse to obey it. It is conscious, it is helpless, and it bites. How does a fungus hijack a muscle? In 2019, the Hughes lab found the answer. The mandibular muscles of an infected ant do not relax. They hypercontract, sustained, violent contraction. So intense, the jaw cannot let go even after the ant dies. The fungus appears to secrete chemicals near peripheral nerves and muscles. Guanobutyric acid, sphingosine, neuromodulators that interrupt the local signaling between motor neuron and muscle fiber. The fungus does not need the brain because it built its own nervous system out of fungal threads, and every detail of where the ant bites has been engineered, not for the ant's survival, for the fungus. 25 centimeters off the ground. Why? Because that is where humidity stays highest in a tropical rainforest understory, north side of the leaf vein. Why? Because in the northern hemisphere, the north side gets the most consistent shade. Cool, damp, solar noon. Why? Because by the time the ant dies and the stalk grows, the conditions for spore release will be perfect. 94 to 95% humidity, 20 to 30 degrees Celsius. The fungus has chosen a microclimate of its streams. The ant is not a victim of a parasite. It is a delivery vehicle, programmed to die in a precise location, like a self-driving car that crashes itself at the right address. And this is not new technology. This is not something the fungus invented yesterday. We have a fossil, a single leaf from Germany's Messel pit with the exact dumbbell-shaped scar that an infected ant leaves when it bites and holds on. That leaf is 48 million years old. 48 million years. The same death grip, the same precision. The fungus has been doing this since long before primates existed. Before humans, before mammals had fully diversified. This is older than us, so much older. Now, here's what scientists really want you to understand. The zombie ant fungus is not unique. Parasitic mind control is not a one-off. It has evolved over and over and over again in different kingdoms of life because it works. Meet Toxoplasma gondii. It is a single-celled parasite that lives in cat intestines. To get there, it needs to be eaten by a cat. So, how does it manage that? It infects rats. And in the rat's brain, it does something extraordinary. It removes the rat's fear of cats. Sometimes it goes further. It makes the rat attracted to the smell of cat urine. The rat walks toward its own death. Toxoplasma boards the cat. Mission complete! This parasite also infects about 1/3 of all humans on Earth. 1 in 3 of us is carrying it right now, including, statistically, some of you watching this. Most people show no symptoms. Some studies hint at subtle behavioral changes, slower reactions, more risk-taking, but the science is still unsettled. Now meet Sacculina, a parasitic barnacle. It finds a crab, injects itself into the crab's body, spreads tendrils through the crab's nerves, and shuts down the crab's reproductive system. The crab is castrated. Then Sacculina makes the crab grow a brood pouch, but with Sacculina larvae inside instead of crab eggs, and the crab cares for them, grooms them, aerates them. The crab thinks it is being a good mother. It is feeding the species that destroyed it. Now, meet Leucochloridium, a flatworm that infects land snails. It grows green, pulsating brood sacs inside the snail's eye stalks. They throb 40 to 75 times a minute. They look exactly like caterpillars. The parasite forces the snail to climb high into bright sunlight, exactly where birds hunt. A bird sees what it thinks is a fat caterpillar, lunges, snaps the eye stalk off. The parasite reaches the bird's gut. Done. 3 completely different lineages, 3 completely different methods, the same brutal idea. Turn another animal into a vehicle for your own reproduction. If this strategy has evolved this many times, you should probably ask, how often is it happening right now, in how many species, and, uncomfortably, what is happening inside you? Here is the part that should sit with you. For most of human history, we imagined the self as a single thing, one body, one mind, one self. Modern science is taking that picture apart. You are not one organism. You are a community. Your gut alone houses trillions of bacteria, more cells than your own body has. These bacteria produce neurotransmitters. They influence your appetite, your mood, your sleep. Studies in mice have shown that swapping gut microbes can change anxiety levels and even social behavior. Most of these passengers are partners. They help you digest food. They protect you from invaders. They are mostly on your side, but not all of them are partners. Some are simply along for the ride. Some, like Toxoplasma, may be nudging you in ways neither you nor they fully understand. The zombie ant fungus pulled the curtain back. It showed us in shocking biological detail that one organism can pilot another without its knowledge, that the body and the will can be separated, that an ant can be aware, see clearly, and walk to its own funeral. You are not an ant. You will not bite a leaf at noon. But the next time you reach for that second cup of coffee, the next time you feel an inexplicable craving, the next time your mood shifts for no reason, pause for 1 second and ask the question that science is only beginning to ask. How much of me is just me? A fungus learned to drive a body. It did it 48 million years ago. It is still doing it today on a leaf 25 centimeters off the rainforest floor at solar noon, and we only just figured out how. The universe is stranger than we think. Sometimes the most alien thing on Earth is not in the sky. It is under a leaf 6 steps from your trail. Next time you walk through a forest, look down, not up. Most of what is happening on this planet is happening close to the ground, between species you cannot see, in arms races older than humanity. And speaking of organisms older than humanity, There is another one I cannot stop thinking about. A creature so small you could fit thousands on a fingernail. It has been boiled. It has been frozen to almost absolute zero. It has been exposed to the vacuum of outer space for 10 days and survived. When conditions get bad, it doesn't die. It dries out, curls up, and waits. Years. Decades. Some scientists think centuries. Then a single drop of water lands on it, and it walks again. Its name is the tardigrade, and the secret to its near immortality is a state of being almost no other animal can enter, a state called cryptobiosis. How does something that small refuse to die? That is the next story. For now, keep looking. The strangest things on Earth are not waiting to be invented. They are already here.